All right, let's go ahead and get started with session 3, which is being chaired by Hana Olsman from UCSF and Michael Reiser from Janelia.
Hello, am I on?
You are.
Okay, excellent. Okay, hi everyone, and welcome to our session on control theory and systems biology. I'm Michael Reiser, happen to be another fly neuroscientist and a group leader at Janelia. I'm an enthusiast for control theory, which is why we've organized this session and put it into this workshop. We'll be touching on many of the common themes that you'll hear about in the other session, but there's one important difference, which is everyone in this session has some background in engineering, and they're going to use the tools of engineering to look at biological systems. And the reason it's important, and I think really needs to be brought into this 4DCP conversation, is that so much of biology is comprised of these loops of interactions, and there's feedback within the loops and across levels. And these systems are very counterintuitive often, but there are actually mathematical tools, some mathematical tools for addressing and working on systems like that. And that's what we hope to highlight in this session. And I'll just say, if people want an introduction to the whole general topic, I could highly recommend Hana Olsman's very recent review article on this, which is terrific, a really nice introduction. And with that, I'd like to hand it over to Hana to introduce the speakers for the session.
Hi, everybody. I've been looking for this session, forward to the session the whole month, actually. So let's get started. So our first speaker is Domatilla Del Vecchio, who is a very old friend of mine from back from the Caltech days. She's a professor at MIT and has done a really seminal contribution to how we think about modularity and signal integration and so on in biological systems. So take it away, Domatilla. You're muted.
Okay, thank you very much. So can everybody see my screen? Okay, so I would like first to thank Michael and Hana for inviting me here to speak. And one point that I will make right away is related to the conversation that was just underway before, this tension between simpler model and more complex model. I will say that a lot of the research that has been driving, our efforts in the past several years has been started by this tension between me, which is an engineer with a reductionist approach, okay, that's try to simplify things to the most, and a dear colleague of mine at University of Michigan, Alexander Nympha, who is a biologist. I was instead pushing for big models that include all the details. So anyways, this talk is about the problem of signal propagation in biology. And the way that I'll start is talking a little bit about the concept of modularity. Okay, so I am an engineer, and modularity for us is critical because describing a system as the input-output composition, right, of simpler subsystems helps us overcome the complexity of analysis and especially of design. So in engineering, we often forget about the details within subsystems when we compose them and just focus on inputs and outputs and connect them, forgetting essentially what's inside the system. But the reality is that we can do that because feedback compensation is used everywhere in subsystems to ensure that they are somewhat robust to their context, so they are somewhat isolated, okay, from all the interactions that we want to disregard. So there is a notion of modularity, right, that has been studied for a long time in systems biology by seminal works such as Alon, 2007, and a beautiful book by Christian and Gerard that talk about conserved core processes, right, through the evolution. And so the question is, can we actually perform this type of analysis of describing a complex system, right, from the composition of simpler subunits, right, that we have characterized previously in isolation? So that's the question. Of course, being an engineer, my answer to that is that, sure, we do that all the time. But then if you talk to my colleague, a biologist, says, no way, why would you be able to do that? And the reality is that this isn't true to a great extent in many situations, right, in biology. And so when we started working on this type of problem, we were working on this genetic activation clock, which is a prototype of the design principles of oscillations in circadian rhythms in many biological systems. And one question we were asking, if you have this clock that is oscillating and now you view that as an upstream system and you want to connect it to a downstream system, is that still working? And the answer is unfortunately not. Okay, so what happens is that the addition of a downstream system, it's interacting in some way with the upstream system that you view as a signal generator. So the information is not going through because of an interaction between the upstream and downstream system that you are not capturing. So the idea is that there are loading effects, right? Anytime you view composition of the system that tend to change the upstream system behavior. So with my colleague, Eduardo Sontag, a while back, we decided to try to establish a control system theoretic formulation of this issue of loading that will capture this notion in biology through this retroactivity signals that capture, for example, the retroactivity to the output is capturing the fact that anytime you connect the system to a downstream system, you have a loading effect that is perturbing, right, the information that the upstream system is sending. And the same is true, right, for this signal here, which we call the retroactivity to the input. The system is always changing any information it's using, right, from the upstream system. And this is somewhat related to what in physics is called the observer effect, right, that you can't quite measure exactly, right, the quantity because you're changing it in the process of measuring. But in any way, so in this theoretical formulation, we will be able to interconnect systems by ensuring that there aren't internal state variables that are shared. Okay, so that's critical. So that said, so we use this formulation and revisited a bunch of models, right, of genetic systems, signaling systems. We focus mostly on genetic networks, but I'll point out also some work that has been done by other colleagues, beautiful on signaling systems. But essentially, what are implications of this loading in genetic networks? Well, the implication is, for example, if you have a gene transcriptional component and you have downstream target sites, these are affecting actually the behavior of your system. You're just changing the temporal dynamics, okay, of the system. So if you look in time, the temporal response, either going up or down, is definitely changed by the presence of any downstream targets that you want to have. So it is also true that if you look at the independently regulated genes in the cell, whether it's bacterial or mammalian, they always have upstream, in quote, regulators, which are RNA polymerase and ribosomes. So what happens is that if you are activating one gene, another gene will always respond, right? Even if you don't view them as connected by a regulatory path. The way to view this is that they have both an upstream node, there is a retroactivity that is going back, so information is really going up and coming back down. And the answer is that this gene here is responding to a perturbation applying to this gene here, even if regulations are going downstream. Okay, so this is just experimental data showing that as you're activating this gene, the other gene, being GFP in this case, is going down. So that said, this can create actually very interesting network effects, right? So if you propagate these issues, right, of having essentially the genes in the cell many times having all these upstream nodes that you don't represent in your model descriptions, but they are there, you're really changing the overall behavior of your network. So what I'm showing you here is just an instance of what can happen, right? So this is a simple genetic activation cascade with 3 nodes. And then you can have essentially these hidden interactions that appear because anytime a gene activation happens, there is sequestration for all these global regulators. So what happens is that you can predict with a model in parameter space what's going to happen to these hidden interactions being stronger or weaker. And you can probe this through genetic constructs, right, built to understand in order to essentially show that you can tune the strength of these hidden interactions, right, by DNA copy numbers and ribosome binding site to essentially tune the response of your system, essentially the way you want. So this is just a way to show that many times when we trying to understand the dynamics or the behavior of these networks, right, the presence of these regulators that are always there are going to affect, right, the behavior of your system. And of course, I am an engineer, so I'm using this for engineering things that behave the way we want, but you can view this also for understanding, right, or interpreting results that you get from natural systems. Another example, which is very intriguing, is one where you have a repression cascade, for example, right? So you have a gene repressing P1, which is repressing P2, but anytime this gene is repressing its target, it's freeing up resources. So there is a self-positive feedback loop that's appearing here. So what that gives you, it gives you bistability. So essentially the simple repression cascade, what becomes a bistable system. And this can be predicted by a simple model that includes this global upstream regulator, and experiments essentially confirm that. So this is what happens, just to give you a sense, in genetic networks, but I also want to point out the work of others. And there is also a very recent PLOS paper that came out a few weeks ago from other groups that are looking in signaling systems, right? So these are cascaded, for example, covalent modification cycles. And what they have shown is that the presence of downstream targets, so for example, if you have your MAPK cascade that has multiple stages, the last stage, right, which has having all these substrates that are targets of the MAPK cascade, are actually changing the behavior of all the upstream stages. And what they've shown, for example, is that the presence of these stages is really changing the characteristics of the steady state response from ultra-transitive down to Michaelis-Menten type. And they're also changing essentially the temporal response of the system, with an overall result that downstream systems tend to decrease the boundary, so the speed, okay, of upstream systems. But that's sad, so I'm happy to point you to more references in that direction. There is a rich group of people. I mean, there is, for example, Schwarzman at Princeton, who is a physicist who is doing, has been doing fantastic work on trying to understand this interaction in signal transduction networks. But the essence of this that I wanted to get at is that mathematical problem formulations, right, inspired from control systems, can help maybe reveal some new biological insights. So when we started from this notion of systems with input and output and back action, our idea is because we wanted to build systems for synthetic biologies that are robust to this loading effects. But then, of course, in the way to try to do that, right, you also understand and discover, right, these mechanisms in experimental system. So this also brings up the issue, this is an ongoing conversation with many colleagues of mine, of identifying intended versus non-intended connectivity, right, in biological networks, right? So when we identify a regulatory link, right, from data, are these really true regulations, or maybe they happen because your nodes are sharing some enzyme, right, or a protease, or an RNAP, or a ribosome? And then the other question, which has been going on, right, is being this critical question of whether biology uses retroactivity and its propagation backward for designing systems. Because there are many situations, right, for example, where you have transcription factors that have so many binding sites on the chromosome that do not have a regulatory effect. They apply a load and they change the dynamics of the transcription factor. So there is this ongoing question of whether biology has done that on purpose, right? Can we demonstrate in any of these cases that actually this downstream loads are there for tuning, right, responses? Of course, from engineering point of view, we view them as something to defeat because we like to modularize things, but biology may use that to optimize behavior. Anyway, so I think I'm already over time, so I'll close there and open for questions.
Okay, Domatilla, can you unshare your slides, please, so that we can all see each other? Okay. I have no idea how much time we have. We have one minute, around 2 minutes. So maybe I can ask a question that probably will get me in trouble. So there's something that you said about whether it's true or just incidental regulation that I think is it's a very important point, because the way we, for example, from I don't know, transcriptomic data or genomic data or whatever, the way we extract pathways and program is by looking at covariation in some way or the other. And that, you know, what you're saying is that that could be very, very confounding. Is that what you meant to say? And can you elaborate on that?
Okay, so I don't want to get myself in trouble, but there are situations, right, that, for example, if one is not careful of taking the correlation, right, and verifying whether molecular interactions are there, for example, gene 1 regulating gene B, while in fact, right, gene 1 is just sharing, for example, a microRNA, right? And there are beautiful papers that show that you can get correlation between two genes just because they are sharing a microRNA. So I'm just saying that it will be nice to have a systematic way to remove those confounding factors when we perform network ID.
Okay.
I haven't seen any other questions. I have a kind of a superficial question, but one that always bugs me is about the overuse of block diagrams, right? Because I mean, for biologists specifically, right? They're sort of everywhere, and you know, I think often engineers use them with the assumption of no loading, right? High impedance interfaces between modules, right? But as you show, right, you have all this retroactivity. So it seems like just conceptually, the metaphor is just bad, right, of drawing boxes around things. So how do we deal with that? What do you think about that?
Well, believe it or not, but if you look in control theory, that has been an ongoing conversation for maybe 20 or 30 years since essentially the inception of block diagrams, right? There's a beautiful work done by Jan Willems, who has been arguing all his life, he's a control theorist, but he's arguing all his life that these are misleading, because they're not capturing things such as back action, right? If you have a motor, you have the back EMF, they're just not capturing it the right way. So I think that most of control theories today, well, many of them will respond that all those effects that you don't like, you put them in a big uncertainty delta around your system, right? And try to fight against it, which makes sense if you want to engineer a system. Probably, but if you want to optimize, co-optimize subsystems together, which is what nature may be doing, maybe not.
Yeah, so Domatilla, the theory that Jan Willems has been kind of advocating is called behavioral systems, right?
Yeah.
And I think it's an area that we all would benefit from looking into for modeling biological systems.
Yeah.
So.
Absolutely.
Great. So if there are no more questions, thank you, Domatilla, for, as usual, a wonderful talk. And let's move on to our next speaker. And that would be Marcella Gomez. Marcella, are you here? Yeah, and Marcella is a rising star faculty in applied math, I believe, at the University of California, Santa Cruz. Go ahead, Marcella.
Okay, I hope you guys can see the presentation mode.
Yes, we can. Go ahead.
Okay. Yeah, so I'd like to talk about what can an engineering approach do for our understanding. And I like to start with one of the quotes that people love to quote. What I cannot create, I do not understand, by Richard Feynman. And, you know, and I wanted to break this down a little bit. What did Feynman imply, or what did Feynman imply by create? Does that mean we must be able to build a cell from scratch? It's notoriously difficult to predictively model complex biological systems. And we could try to create a mechanistic model from the ground up and model every single interaction. But we know that system identification is hard. These systems are partially observable, and they're high dimensional dynamical systems, which would require a lot of experiments with sufficient dynamics, because we need persistent excitation in order to uncover all the parameters. And then even if we can achieve this, can we still gain understanding of universal principles, which cannot necessarily be extracted from a high dimensional mechanistic model? And so what does it mean to understand? There's different levels of understanding, and what can be gained from each level, and how much understanding is enough to move forward. And so I'm an engineer, and engineers can create, or we like to think we can create with sufficient understanding, an example being the field of synthetic biology. And is sufficient understanding achieved when we can control a system? And so I'd like to propose a potential modeling framework and control theoretic framework to tackle this problem. And so for that, I'm going to start with, I'm going to share an example of a research project I've been working on for the last couple of years. And the challenge has really been that before I started this project on wound healing, controlling wound healing, a lot of my focus was mostly in modeling gene networks in E. coli. So going from modeling at the single cell level to modeling complex tissues and biological processes really forced me to take a step back and try to look at this problem from a top-down approach. And so our goal here is to accelerate wound healing by reducing the inflammation stage. And so wound healing involves this carefully coordinated symphony of biological processes. And so we really need to simplify it as much as possible in order to achieve our goal. And so we focus on macrophage recruitment and macrophage polarization or activation. And so M1 macrophages are primarily affiliated with the inflammation stage, they clean up debris, et cetera. And then the macrophage 2, M2, are associated with proliferation stage. And the biggest challenge here is defining the control objectives to achieve our goal. So how do we map actions that we take at the single cell level to tissue level response? And for this, I'll propose a model-based systems theoretic approach. And so we start with a hierarchical model here. So we can drive systems without a comprehensive model. So what is the right modeling framework for understanding complex systems? Different levels of abstraction are useful for different questions and make use of various data types. But a hierarchical model could potentially make use of time series data and observable states at various spatial and temporal scales and allow us to map across different scales in order to understand complex processes. In this way, we can maintain preservation of truths and consistencies. One thing that is very well known are the 4 stages of wound healing. And we know there's inflammation. Inflammation is always followed by proliferation. And so at the highest level of our modeling framework, we can incorporate this. And for that, we can use different types of data-driven models. We've looked at classification algorithms for modeling wound healing stages. At the lower level, if we want to then map from single cell activity to this higher level abstraction, we can apply a mechanistic model. And these states can also be informed through data in terms of like how many states can we reduce the model to, and also we can search the literature for parameters. And at the lowest level, we really care about being locally predictive, right? So we don't care about having a qualitative model that preserves universal truths, but rather are predictive enough in the short term for us to take immediate action for control. And so this helps us map single cell activity up to the tissue level response. And so for the top part, we use this information and modeling framework as a part of what we call the decision maker, which I'll explain later. And then the lower 2 levels of modeling for a planner. And so we'd like to take a hierarchical approach where we have our decision maker, which is a high level model of the wound healing process used to identify wound healing stage and timing for intervention. The planner, which is what we use to plan the intervention strategy, and which essentially is generating a reference signal for our low level controller. And so the low level controller is then tasked with executing the intervention strategy. And so I just wanted to go through an example of what those models might look like for each of those stages and the different types of data sets that we make use of. So for the decision maker, we had a lot of transcriptomic data. So I'm kind of like a scrapper. So I essentially just search the literature and just try and find whatever data we can make use of, because the generation of data is really expensive and time and costly. And we needed results pretty quickly. So we found some transcriptomic data. At first, it seemed almost impossible to extract any ground truths, but eventually we were able to find a method by which we were able to map across different data sets from mouse to human, from burn to acute wounds. And essentially using a 5-dimensional state, expressing relative gene expression over time, we were able to then train a support vector machine to then predict wound healing stages based just on gene expression with 58 identified biomarkers. On the image side, because we were working with whatever data we could possibly get our hands on, we also had these images of mice. And these are not really used or quantified in any way, but rather are kept just for as part of the protocol for visual validations for the clinicians. But we actually worked on building a survey to annotate these images, and then trained a convolutional LSTM network to predict the wound healing stage based on the images. And then we were able to find a data set that had both gene expression and images and able to map the two together. And so this serves as a sort of validation. So here we have the image-based model assignments. These are probability distributions of being in any wound healing stage over time. The x-axis is days, the y-axis is a probability from 0 to 1. And so in the beginning, we have hemostasis, then it's followed by inflammation. You can see we have proliferation, and then a really delayed uptick in maturation. And if we look at the gene-based model assignment, it's a slightly different mapping, but it seems to make some sense. So we have our inflammation is assigned here at day 1, but proliferation is assigned much earlier here at day 2. But it makes sense that, you know, the gene expression for proliferation would be upregulated much earlier than we would see visual cues, right? So remember, one is image-based and one is gene expression-based. So finding that mapping is another challenge. For the planner, on one end, we tried a mechanistic model of wound healing. So because the gene expression data told us that we can reduce transcriptomic data down to 5 dimensions, and that was enough to uniquely identify a wound healing stage, then it made sense that our mechanistic model, phenomenological model, should sufficiently describe wound healing, at least for the purposes that we care about, with 5 states. And some of the parameters were gathered from literature, and we also did some data fitting, again, to these images of time series of wounds on mice by coming up with an automated method to track wound size as a function of time. And then we essentially use this model to determine most effective treatment times for macrophage polarization. So what's the best time to start the treatment and how long the treatment should last, identifying the effective regime, dangerous regime, and the non-effective regime. And then we came up with a modeling or locally predictive model of cell migration into electric field. And this is more pertains to the task of recruiting macrophages. And so we have cell migration under an electric field for different EF values. We do single cell tracking, quantify the directness, train a recurrent neural network, and we're able to predict the movement of cells under EF. This model also works offline, so we can do some planning of optimal EF signals to recruit macrophages with minimal effects on the morphology or changing phenotype of the cell. And so finally, we can use those to inform our low-level controller, and the low-level controller can make use of the model, but we can also do a neural network-based model in which the controller doesn't necessarily need to have any information of the system it's trying to control. And so we were able to demonstrate this with feedback control of stem cell memory potential, in which we had this RBF-based neural network that was not trained offline or it required 0 data, was just initialized randomly. This controls the external concentration of protons, so thereby influencing the extracellular environment and the pH. And this in turn affects the memory potential of the cell. And so then we were able to show that through the RBF neural network-based controller, we could then drive the potential membrane towards a given value, which we then hoped would drive the system towards a specific cell type and differentiation. And yeah, so that's the cumulative summary of the approach we've been taking to tackle this endeavor. And this has been a highly collaborative project with clinicians, biologists, and people who do bioelectronics. Thank you. Sorry, stop share.
Okay, thank you, Marcella. So I'm going to take the liberty to ask a question, and please, the audience can chime in. I don't know how much time, the timer hasn't reset. Okay, we have I think the timer is going a little berserk.
Hana, the
yes.
timer counts up to 5 minutes. So you have until.
Oh, it does
5
count
minutes.
after 5 minutes.
Yep.
Well, okay. Okay, thank you, Janine.
Sure.
Okay, so Marcella, you described to us a kind of a successful round of modeling at these different levels, and then the ability to steer the cells and differentiate them the way you want them to. Can you actually dig a little bit under the hood and tell us where that process, which integrates so many scales, I mean, it integrates, macrophage biology to 5 states, 5 variables at some level. Where did that go wrong? Where there failure modes in that integration?
Oh, sorry, I wasn't clear. So for the stem cell project, that was just an example of doing direct feedback on the single cell level. But then the idea is like, how can we scale up that idea to a tissue level? And so far we've been able to at least try to come up with models at different layers of the hierarchical model, and trying to draw consistencies and mappings across them in the hopes that when we manipulate at the single cell level, it will propagate to some response at the tissue level, being a shortened wound healing time. But I would say, really, we need those validation experiments, right? This is sort of building just the theoretical framework to move forward and then inform the experiments. But we do eventually need the clinicians to go, do these dynamically timed therapies and then visualize the response of the mice. And it has to be something that we can visualize. There has to be visual indicators that we can monitor in real time to then inform therapy in time as well. So that's one of the biggest challenges, because we cannot do like I'm not sure if that answered the question.
Yeah, it does. So we have a few seconds left. Anybody wants to jump in and ask another question? I actually do have a lot of questions, but I'll keep that to the end when we have this discussion, with all the speakers. So thank you so much, Marcella. Beautiful work. Okay, our next speaker is Noah Olsman, who is a postdoc in the lab of Johann Paulsson at Harvard. Go ahead, Noah. You're muted.
Unmuted. Yep, great. Thank you, Hana. So I'm going to talk to you about the perspective of someone who's a theorist in the last 2 years who's moved in doing experiments, and talk to you a bit about what I've seen as trying to really do experiments motivated by theoretical results, and hopefully give some ideas that connect to some previous things and talk about how we can sort of use theoretical tools to actually design experiments rather than just analyze everything about what's going on. So I'm going to tell you about sort of a subsection of a project I'm working on, which is to try to look at single cell induction curves. So actually look at like a single cell turning on gene expression, understand the noise and dynamics and variability in that process. So the basic idea here of the experiment was to say, okay, I'm going to add an inducer. I'm going to turn on a sigma factor. So this is an E. coli, this can be a bacterial transcription factor. And that sigma factor is going to turn on a GFP. And oftentimes we sort of see these things as a dose response curve, maybe with some error bars, but we really don't actually have a good sense for what did the single cell kinetics of gene induction look like in a very precise and sort of theoretically grounded way. And so the basic idea of the experiments we were trying to do was to say, okay, let's look at fluorescence over time. I'm going to give a linear ramp of an inducer, and then I'm going to vary the ribosome binding site strength to essentially change the rate of induction of the circuit. And so the idea is the weakest ribosome binding site should have a relatively weak induction. And then as I increase the ribosome binding site strength, I expect to get faster induction that's stronger and stronger. And the real hope was that we could actually start to parameterize and understand induction systems in a model-based way using data, not just sort of getting qualitative ideas of it, actually, but understanding the kinetics and timescales. And so my collaborator, Jacob Shenkar, and I worked on this, and we managed to actually get this experiment to work. And we're using single cell microfluidics to study these. And while we managed to get these to work, we got induction curves that look, pretty good. We see that there's a regime where it's, a weak induction. We see there's a range of induction curves, and we see that there are these very strong ones that saturate very early and induce very quickly. But if you look, there's two things that sort of pop out. One is, while this weakest induction curve is quite, consistent and low noise, the stronger ones are both noisier, but they're also really varying in time. And I think a lot of time when we look at biological experimental data, we just assume there's going to be some amount of variability we can't capture and understand. And we're saying, okay, sweep it under the rug, it's part of biology. But when we started looking at the actual data, we started noticing some weird things. So one example of this is we found that when we look at the coefficient of variation, which is basically the noise in the response relative to the mean, or as a percentage of the mean, we saw that at low induction levels, we had relatively low noise, but in some of our stronger circuits, the noise was actually increasing with induction strength. And this didn't really match with any theoretical models we had or the ideas we had about how the system should work. And so we decided, rather than sort of saying, oh, this is biology, we don't understand it, we wanted to actually see, could you actually get these kinetics to look like what the theory says they should look like? And so I'll show you an example of what one of these experiments looks like. So this is a device called the mother machine. Right now, this is going to be dark, and then basically we're ramping up induction. You're going to see this, these cells, gradually get brighter and brighter and brighter. Each cell is in a single trench growing in an isogenic population. We can grow these cells for days at a time, so you get really long-term induction kinetics. And they're seeing this linear ramp of induction. And so this is for one of these weak ribosome binding sites. We saw the induction looks pretty consistent. The cells don't look all that different. They're sort of turning on a consistent time scale. It seems fine. There's some cells that die, but that's kind of all experiments. But then when we went to the stronger ribosome binding sites, we see some pretty weird things. We see a lot of spatial variation, so from one trench to the next. But we also see a lot of population variation. Cells will get bright and dark and bright and dark. And we didn't really understand why this was happening. We saw this thing where we're seeing these fluctuations, and it seemed really not to make any sense, given that all we're doing is turning on a GFP. And I'll zoom in a bit and sort of show you what we see here. And when you look closely, you'll actually see we're getting oscillations and we're getting plasmid loss. We're seeing cells just go dark. And this, I think you could write this off as noise, but this is a really kind of weird phenomena to happen in just such a simple system. And so as a control theorist, there's actually some good theoretical ways of thinking about this type of behavior. This stuff there is actually indicative of certain types of problems in a control system. And so the first question you'd ask is, what is a control system we're talking about? Well, all of these circuits are on plasmids. So just like Domatilla mentioned, there are these things with DNA copy numbers, and especially in synthetic biology, we often express things on plasmids. And these plasmids, the way they work is they have to be continually reproduced as the cell's divided, or else you can run out of plasmids. And to make sure the plasmids don't go too high or too low, there's copy number control via negative feedback that is built into these plasmids. So, even though our circuit is just a simple gene induction system, at some level, we are actually building it on top of a feedback controlled system. And this gets to what sort of Hana said, respecting feedback loops. And so I'm going to talk. So the question is, why would we expect, if there's feedback control, why would you expect this to have anything to do with the variability in our system? Shouldn't that just suppress the variability? And actually, Dan Fisher mentioned this idea, a canonical example of feedback control in biology, which is this bacterial chemotaxis system. And this is one of these first examples of what we call robust perfect adaptation. And I think a lot of time when people talk about robustness in biology, they're talking about steady state robustness. You're adapting back to some constant level. And here you have a single cell, there's some intracellular signaling marker, there's disturbances, and you see there's a transient effect, and then it adapts back to the same level. You get this nice adaptation. And so this was actually in a paper that my grad school advisor, John Doyle, wrote 20 years ago that showed that this robust perfect adaptation was actually an instance of a well-characterized version of feedback control called integral feedback. But one thing that we learn, I guess, like the tools of control theory give you, is that there's more to robustness than just adapting to a steady state. Robustness is about dynamics in a lot of ways. And so the way we can quantify that is, rather than just saying, what is the steady state error, or something like that, we can talk about this idea that actually Tim O'Leary mentioned, which is the sensitivity of a system. And the sensitivity basically says, how big is my error relative to the change in the input to my system? And so you can think a sensitivity of 1 means you're basically doing as what, you're doing as well as you would if you didn't have feedback control. A sensitivity less than 1 means you're suppressing errors, and a sensitivity bigger than 1 means you're increasing errors. And we can write down the sensitivity function that quantifies, as a function of time varying inputs, say the frequency of a sinusoid that you're giving to a system, how well it's tracking the system. And so one thing so as an example of this, you can see in the bottom left, we have really like low sensitivity, which means we're tracking this low frequency signal really well. The blue and the gray curves are almost on top of each other. As we get to a higher frequency, we see a little bit of mismatch, but it's pretty good. Then there's a peak where we're actually perfectly mismatched with the system. We're going left when we should be going right. And then at sufficiently high frequencies, you're not even really adjusting. It's like if you're like moving your steering wheel back and forth super fast, you're just not going to actually respond. So what sensitivity? So the question is, why wouldn't you just always make the sensitivity really small for all these systems? Why would you have any of this be non-zero? Well, also, as similarly mentioned, there's this guy, Hendrik Bode, in the 30s, who developed a lot of theory around this. And one of his theoretical results is that of Bode's integral theorem, which basically said the area above this line of like null sensitivity has to be bigger than the area below. And so this is like a conservation law for sensitivity. If you want to suppress sensitivity in one regime, you have to increase it somewhere else. And this is a fundamental result for any feedback control system. So what this tells you is that you're always paying a price. There's a trade-off. And one way of talking about this trade-off is the fragility. So how bad your worst case sensitivity is versus how fast you respond to a system. And here you see we have this blue curve, which is really, really fast, but it's really unstable. It's oscillating wildly. Then you see this purple curve, which is like really slow and steady. It's not quickly filtering out like the baseline effects, but it's not rattling around a lot. It's not responding to high frequencies really badly. And we can sort of see these as just parts of the sensitivity function. So if I do the sensitivity function for each of these points on this trade-off curve, the purple one doesn't filter out signals well, but also doesn't amplify them. The blue is really good at filtering out low frequency signals. It adapts really quickly, really well, but it also is really sensitive to high frequencies. And so it turns out when you turn up the gain of a feedback system, that often makes you more sensitive to high frequency signals. And that causes you to start doing these oscillations and go towards instability. Well, if we think about this data I just showed you, we saw that these cells started to oscillate. And it turned out it was only when I had strong gene induction. And I sort of used this control theory tool to tell me, oh, this looks a lot like I'm screwing with the sensitivity of my feedback control system. And it turns out that's exactly what the problem was. It turns out that the problem we were seeing was entirely caused by the fact that our induction system was messing with the control loop that controlled the plasmid. So it's turning the gain of the plasmid copy number control way up and causing the system to become unstable. And so why do we get plasmid loss? Well, if you're oscillating around a bunch, eventually you're going to hit zero. Once you've hit zero plasmids, you can never get them back again. So it turned out this understanding of feedback control actually told us how to make really consistent gene induction systems. And then we fix this problem, exactly the same, all the genetic parts. And now we get extremely, extremely consistent induction with these, with exactly the same system. And so this told us you really, really could make things more precise. And to show this quantitatively, well, here we have those original induction curves. Here's what happens when we fix this interplay between my induction system and the feedback control of the plasmid. You get these curves that have essentially the minimal possible noise. And this is real data. This is real single cell data. And you can get what look like plots from a differential equation. And this sort of, I think, is a good example of the fact that you can actually make biology look really, really predictable if you do this thing where you understand how the systems you're studying are interacting with feedback loops and know how to mitigate the effects of your measurements, sort of in the same way that Domatilla mentioned, with the systems you're trying to measure. And so I think one story you could tell is that, oh, this theory helped us understand the experiments, but also the experiments helped us do the theory. Imagine trying to do this if all you had was, say, plate reader data or flow cytometry. You would never see those oscillations. You wouldn't see that dynamics happening. Really, the core thing that let us do this was both the theory, but also this single cell microfluidics let us see at this very high time resolution what was going on in our data. And I think this is, if there's a takeaway for Janelia, it's that understanding the relation, building the tools to let you think about the theory can really push both the theory and the experiments forward. You can't really use the theory if you don't have tools that can actually give you the data you need. And the data can tell you what type of theoretical tools you need to develop. And I think for cellular physiology, we really, really need to figure out what are the tools we need to make the experiments really, work, and also to make the theory useful. And so with that, I want to thank Jacob Shenkar, who's a grad student in our lab, who's helped out with this project a lot and worked on it, and Johan Paulsson, who's my advisor. Cool, thanks.
Great. Thank you so much, Noah. Any questions for Noah? As usual, I have like a billion questions, but I don't want to dominate the conversation. Okay, I'll ask my question then. So Noah, I'm a believer in this, right?
Yeah.
So I believe that you kind of, when you mess around with a feedback loop, the consequences are severe. But this is one example in one very simple induction system. Can you elaborate a little bit on how you think about that, tackling, I don't know, a developmental program, a whole pathway, a cell, a mitochondrial dynamics, all of this, kind of beautiful phenomenology we heard about all through the day-to-day of very complex systems.
Yeah, and I think that's where it gets tough, but we do know that these things are all regulated by feedback control. There's homeostasis in every cell we've ever seen that exists. And I think that while it's going to be much harder to do, like we can start getting dynamic. I mean, your lab has done plenty of single cell dynamics of long-term experiments in mammalian cells. Like we really can actually do these perturbations. And I think a lot of the issue isn't so much like having some really simple, precise system, but having a system where you can understand how it is that, the thing you're doing to the system in the experiment is affecting the physiology. I think that's kind of one of the core tool development things that control theorists can bring, is say like, look, how is it that this measurement I'm doing, like Domatilla said, is affecting the core feedback? And if it's affecting in an unknown way, then that should be a new research direction of like, okay, what is happening that isn't explained by my model of what's going on? And I think that's where the control, the sort of science and engineering feedback loop can sort of kick in.
So we have Binyu has her hand raised. Go ahead.
Yeah.
Oh, you're muted.
You're muted.
Sorry. If I connect your talk with the earlier talk by Domatilla, it's possible that your control, the whole PDE will be changed too, because the network is changing. How would you deal with that?
Wait, the whole what? The control the...
I mean, you have a PDE, right? I mean, I'm not a country series, but if I generalize the earlier talk conclusions that the PDE could also be changed, you have other factors, other actors come into play, that how would you see yourself in the real cell? I would think that will happen. How
and
do
it
you?
Does, and
yeah.
So I think what we see is that cells go to really great lengths to control, like to really insulate the important control systems. And actually John Doyle has this great phrase of like constraints that deconstrain. There are some things in cells that have to be actually really, really precisely regulated so everything else can be sloppy. And I think, one example of this, I mentioned the stat, is like heat shock control. A cell needs a really, really high performance heat shock control system to deal with fluctuations in temperature. And that lets everything else in the cell not worry about temperature very much. But if you screw with the heat shock control system, that can lead the cell to die very quickly. And so I think the biological insight is that we should look at these things. What are the things that are hyper conserved in biology? And that can point to things that are the things that let other things be sloppy.
Very quickly, Noah, we're running out of time. Hernan Garcia is asking, how did you fix the feedback? He missed it.
Yeah.
That's an important point.
Yeah, I didn't get into it because it's a few different things, but the important thing is just do not let any transcription touch the replication control machinery. So we have nothing pointing at the replication control machinery. We use ultra strong terminators. Basically, it was just build in as much insulation as possible from the transcriptional machinery so that the circuit wasn't actually going in and touching anything that's sort of part of the important mechanism. That's sort of this, by adding in that constraint that deconstrained the rest of the circuit.
Great, thank you to all the speakers of this. Hopefully, they gave you a kind of a different way to think about biological organization that you're gonna take with you to all of your other work. Michael, what are we supposed to do now?
I think we are into our discussion, right?
Okay. So, right. So let's start our discussion by seeing if there are any points that either the audience or our panelists here would like to, elaborate on or pose. I think it would be either jump in or raise your hand and I'll call on you. Okay, Herbie, go ahead. Herbie Levine has his hand raised.
Yeah, I mean, I want to go back, I guess, to the talk about the block diagrams and modularity. So we've been working on, interactions between cell decisions about motility and cell decisions about their metabolism. And it's worse than the case that the talk discussed, because we don't even have a natural flow of information. It isn't that the cell only gets signals of a certain type from the outside. It gets signals through the metabolic system, and it gets signals through the signaling system, through the genetic network. And these things have, by our count, they have as many cross-links between those pathways as they have internal links between things that you naturally would have said are metabolic enzymes or metabolites. So I'm at a loss to figure out whether the papers, claiming modularity were just completely misleading when it comes to looking at different systems. And this is not just between those systems. So for example, it's well known that the metabolism has amazing interactions with chromatin modifying enzymes. Many of them use metabolites as cofactors in all the reactions that affect the chromatin structure. So it's not just transcription factors, it's also chromatin factors. So is there any hope from a control theory point of view for thinking about this that I'm missing? Maybe that's that's a remark and a question, I guess.
I can try to address that. So that's a great question. So if I put my engineering hat, so I would say yes, there is hope, because we can engineer things today by explicitly accounting for this context effects and rejecting them by proper feedback compensation that we couldn't do right before. But the caveat to that is that the systems that we're looking at are much simpler, right, than you are mentioning at this point still. Nevertheless, right, we can approach this bottom up and say, okay, what are the interactions that I don't want to model? I know they are there and I want to be robust to, and then I engineer a feedback loop around them so that my system has the nice input-output behavior despite everything else, right? So engineering wise, so the problem is well formulated. So I think there is definitely a lot of hope, and that's what we mostly focus on. The other side of the coin, which is for understanding a natural system, which is so complex, right? So how would you bear this type of approach about? And we have been thinking about that for many years. And so something that we are, approaching as a potential idea, and I'm happy to discuss, is that among all the interactions that you mentioned, there are some for which you have an idea of what molecular mechanisms come into picture. So for those, you may be able to use some physics-based models, right? But then you know that there are other interactions and you have no idea how they play a role. For example, there's this interaction between chromatin and metabolism that you know it's there, but you don't know. And then one option is to put that in an uncertainty that you then try to learn by using data-driven approaches as opposed to model-based. And so one approach that you're looking into is merging, right? These two approaches, model-based for the things that I know how to capture, and data-driven for those was complexities too large to model.
Okay, well, I'll look forward to seeing what happens as you try to move down. For a long time, I thought that that's a underdeveloped area. People either do machine learning or they do mechanistic based models, and it's clear that there's no reason not to combine them. So I'll be looking forward to see if that helps.
Herbie, that's exactly what we're, why we're bringing these, the control theory and the data in the same place, the database methods in the same place, because there is no way, there has to be some integration, and it has to be done well to be productive. So, we have two raised hands, Armita and then Daniel, but Jennifer Lippincott-Schwartz has a related question that I'm going to pose, and then we go to Armita and then Daniel. So Jennifer says, we are in an era of gene editing where CRISPR can be used to knock out, replace, or modify a particular gene. Given the idea of non-intended connectivity of biological networks, do you see this as a problem for the field of gene engineering of organisms?
Yes.
So Noah, go ahead.
Yeah, so I mean, I think that's actually one of the big problems that synthetic biology has really contributed to the science. Is we tried to I think the earliest of synthetic biology was us just like throwing parts together and sort of thinking how they'll work in an intuitive way. And then we, I think everyone who's done the control theory to experiments jump has seen like, oh, wow, everything I think is going to work causes something is, trying to get like the carpet that's too big to fit into the room, like a corner pops up everywhere else. And I think the way this interacts with the science is it means that we need, and to Herbert's question, we need to understand how biology does it. I think that will inform the engineering, is I think we still just don't understand how it is biology doesn't constantly get bogged down by these problems. And I think if we can use the feedback of the engineering to tell us what to go and look for, and then using the biology to say, okay, how do natural circuits do, and what are the design principles? I think that's an underdeveloped area that we're really realizing now needs to be figured out before we can do really important gene engineering.
Great, Arvita, go ahead.
No, just going to add one point.
Go ahead.
No, even just from like relevant neuroscience experience, again, coming in with a control theory perspective, right? The issue is we have a measurement problem, right? Because when we manipulate systems and we only measure the obvious things, right, we can miss many of these network effects, right? And so I think the challenge is, do people really want to do the wrong experiment, right? Do your manipulation, but then measure all the other stuff that is supposed to be irrelevant if the block diagrams were correct, right? And I think in general, that's not a mode of science people are doing, because doesn't seem productive and it's only confusing, right? But that seems like what people will have to do more and more if we just want to even understand the data issues.
Yeah, hi. Thanks for this session. I learned quite a bit. So here are my naive questions, I think, about the control theory approaches to biology. So on the sort of mathematical side of the story, if you were to set up a control theoretical problem, right? So you need to define what your objective function is, what the cost function of the control is, what you want to minimize in your system. And I think Marcella sort of hinted that you're also not really modeling the dynamics in sort of a long term, but build locally predictive models and you sort of modify them as you move forward, right? So now thinking about the biological system and whether or not it's consistent with, let's say, controlled or optimally controlled system, right? It seems to me that one needs to know a bit more about, some dynamical equations, the nature of the noise and things like that in the system. Now, there can be a scenario that, the specifics of the model you put in are not that important, and that would be a very interesting scenario. So are there examples that you have been looking at that the model you're putting in is wrong, but what you're getting at is okay in some sense, and it doesn't matter that you have oversimplified or simplified your model quite a bit. So that's basically what's the recipe of building a control approach to a complex biological problem.
I would say that our 5-state mechanistic model is not something that is accurate, because it's almost kind of like a toy model, right? And somehow we're using it to inform real-time decisions, what you can call precision therapy, right? Because it's going to be dependent on the response of the biology in real time. But it gives us a sort of kind of a vague, well, the other thing is like the thing of optimality in biology, because biology works on very long time scales. Things don't have to be exact. So if I say time the initiation of therapy at the onset of inflammation, if it's plus or minus 6 hours, it actually doesn't make a huge difference in the outcome. And so that provides a lot of flexibility that we don't think about, right? And so the mechanistic model can give us a general idea of the window of time for which the therapy should be executed. And then at the single cell level, we can do predictive control over single cell response, right? Depending on what we're trying to manipulate, if it's a macrophage recruitment or activity. But then at the higher level, where we're identifying wound healing stages and looking at the overall progress of the wound, wound closure, or other features that we might track, this can help inform whether, independent of the accuracy of the mechanistic model, whether we're achieving what we want to achieve, right? And I think another thing that we would like to do, and I mean, there's a lot of undone work, like work that needs to be done still, would be how can we take the mechanistic model and adapt that in real time in order to better match the timescales. Because what we want to preserve, we know that the qualitative dynamics are universally true, right? But in some sense, they're distorted, right? The timescales can get distorted and a little bit shifted for each of the processes. The overlaps can become longer, the overlaps can become shorter. So we need to find a way to be able to adapt that in real time.
Does this connect in some ways to, the discussion in sloppy model session, right? So the nature of the problem is, in a sense, it's in a way that, you can ignore quite a bit of parameters, specifics of the process, right? And can be formulated more sort of accurately and mathematically.
It does relate very greatly. This is why I wanted to kind of bring that up in the other session, because all of these issues relate very intimately, actually, and it's an area that needs a lot of work and could be very productive to push all kind of biology with theory forward. So Daniel.
Yes, I guess I want to pick up on some of the things that Noah said in our meeting and make another point too. So one of the roles of theory, which really hasn't been brought up yet, is to the question of what should one be surprised by, right? And so one example one could say in chemotaxis, which I think the physicists were surprised by how it did the controls that would adapt to be able to work in different concentrations. Well, I think the control theory said, well, that's obvious, it's integral control, or whatever the right word is. And so therefore one shouldn't be so surprised by it. So if one looks at bacteria or tries to understand bacteria, they sort of seem as if the gene regulation of the things and feed and sensing things, they do in some sort of sensible way. And then you look at eukaryotic cells, and I guess even people who work on them will say, well, it looks like they slop this together, they slop that on that, they slop this on this to give more complicated feedbacks, and somehow in the end it all works. It's clearly very sloppy. So what I sort of wanted to ask was about trying to bring together actually evolutionary things with control theory and feedback. So there's a very nice body of work with Paul Francois and Eric Siggio and Vincent Hakim on just trying to evolve various things by starting with random genetic networks and just evolving things in a completely toy way, right? Not claiming to be anything connected with reality, and then sort of seeing what comes out. And suddenly they get things that come out that actually give hints as to how biology might do something and turn out actually to be right in one interesting case. A very non-obvious way of doing it. And other ways to get things out which look roughly like Drosophila development, or at least stage of segmentation and things like that, which I think people sort of find, why does it do it this weird way instead of doing a nice Turing mechanism or something? And so it seemed something which can give one intuition in a different way. And so I wonder whether people had looked at that in the context of trying to understand feedback and things, of really starting with sort of random things connected together, but trying to select on something or optimize in a loose sense something and seeing what comes out.
Noah, do you want to jump in? You're unmuted.
Yeah, I think I can't think of an example of that specifically, but I think to the broader question, I think there's some really interesting, I think the broad question is really interesting in how do you evolve systems like this? And I think it's even more difficult when you think of how do you evolve unstable systems that are stabilized by feedback control? Yeah. So glycolysis uses one ATP to make two ATPs. It's unstable, but there's feedback that stabilizes it. So was the original glycolysis system just an unstable blowing up all the time and it evolved feedback? Or did the feedback somehow precede the metabolism? And I think that's kind of a fundamental question that we actually don't really know. How do you evolve to control unstable systems that kill the cell?
I mean, I think that's one example, a very, very simple example of trying to think of what might have come first. Right. Yes. If you have something which seems hard to evolve with something that comes first, and the natural one there is oscillators, which if you start, which you can easily see there's an advantage to having certain kinds of switches, like day-night switch or various other switches. And then you go, geez, okay, if I think of that as being the basis of what eventually turned into a circadian oscillator, then that's a very natural way. You didn't have to invent this thing that was complicated and had all these very nice timings and things and things in it. So I think it is something which seems in a general way to sort of really push on. At the very least, it gives one sort of stories in one's mind and can help on this question of what's so surprising. And maybe that can really help in actually directing experiments of which experiments are really worth doing and which systems are really good to push on. And I think that's a really important role of theory as well. in many contexts.
So I see 3 raised hands, Genevieve, Ben, and Timothy. And I actually don't know what you want to ask. You want to, do you want to go in a different direction, or do you want to comment on what's already been discussed?
I just wanted to make a quick plug for working with biologists and actually like opening up the conversation more, because I think that if so to give an example from Noah's talk, when he talked about building in enough terminators and insulators, as someone who's been trained in genetics, my brain immediately went to, epigenetic modifications, these long areas of DNA that are built in as insulators that you could easily then generalize and extrapolate to his findings that's very specific in order to see how the theory then relates to a generalizable mechanism. So I just wanted to, basically go back to Daniel's point from earlier about how it's going to be so important to bring in lots of different expertise and make sure that, there's ways to work together and bridge the divide, because I think that is going to be a way that will allow for much rapider progress.
I see a collaboration in the making here, Noah and Genevieve. Okay, Tim, go ahead, please.
My quick question, I hope, was really for people who do these experiments. We hear a lot about getting good measurements, but one of the things that transformed neurophysiology was actually being able to clamp a system at a particular value and break a loop. So you have this horrible thing that's feeding back on itself. It's a nice thing, but it's annoying as an experimentalist or a theorist. And one way to tame that is to hold a value at a particular, hold a variable at a particular value and see how the system responds. And as I understand it, that's super hard to do for a lot of cell biology. So are there tools that are making that easier?
Tim, you should read some of our work on that, on optogenetic feedback control and other.
Yes,
there's some beautiful work out there.
Is involved as well.
But it is not mainstream. It's not being used as it should by cell biologists to actually make it, make it kind of a useful tool as it should. So thank you for making the plug for that. This is something that a lot of people have been trying to bring into the mainstream as an experimental way of pushing a system so hard that it actually reveals its modalities and timescales. So Ben, go ahead.
Yeah, just want to go back to what Noah was talking about, like try to learn from the natural system about how to adapt. And there's a concept from causal inference. Of course, it's not dynamic, called incremental treatment effect. Like, do people in this world actually try to just push the system just tiny bit so that you don't alter the system dramatically and you can still observe the natural progression instead of like see big effect, but then it never happened in nature? Is there such a methodology people try to do? Just, of course, there's a sensitivity to experiment how much you can manipulate, but is it some like people want to do that, try to keep within the natural kind of environment and then see how much things will change more gradually than like induce big changes?
There's a big new tool, which is DCAS9 knockdowns, which lets you start doing that. You can do sort of like subtle changes of like sequestering RNA and get like pretty small disturbances to a genetic circuit and then see how the circuit recovers and does stuff. And like, I think we need more tools to do that, but I think there's increasingly cool molecular tools to do exactly that.
But absolutely, and I think the in the era of CRISPR, I think we're starting to be able to do that, and others tools of synthetic biology, we're starting to be able to do that more and more, which titrate in different and optogenetics and all of that, titrate in more gentle, more gradual, more more subtle perturbations. But then, what Jennifer Lippincott-Schwartz has posed in her question becomes very relevant, right? Because we need to have a framework that accounts for, maybe retroactivity, for modularity, for all of these effects, maybe in our understanding of how these perturbations quantitatively affect or how to use them quantitatively in order to do causal inference in the right, most rigorous way. And with that, we're actually officially out of time. That was really, really stimulating. Thank you all for being here, and thank you to the speakers of this session for tickling our imagination. Okay, what do we do now, Michael? Or Janine?
Yeah, thank you, Hannah and Michael. I think that concludes our sessions for today. I just want to thank all of the speakers for great presentations and the discussion leaders for very stimulating discussion. And I want to thank the audience for joining us. Come back tomorrow. We will resume at 11 a.m. Eastern time with the second half of the workshop. In the meantime, panelists, we have a closed discussion scheduled to begin in about 10 minutes. So I'm going to resend the link for that right now, and we will see you then. Thanks.
Great. See you all tomorrow.